Hi there guys, Mr Phillips here, continuing these videos on AS level biology revision to try and help you at the moment, and we've got here a very strange picture, and I just want you to have a think about what that is. I'll reveal it at the end for you. But today we're going to look at enzymes, how they work and how temperature affects them. So most of this should really be GCSE revision, but obviously a bit more details needed for A level. So we should know, and I'm not going to go over it anymore, but here's an example of a graph showing you how enzymes work. So if you had no enzyme, the activation energy would be the red line there, which shows you need a lot of activation energy. With the enzyme for the hydrolysis reaction breaking maltose into 2-glucose or alpha glucose molecules, you would have the blue line. So you've got this difference here in the amount of activation energy that is needed, and that would mean the enzyme happens a lot quicker, and it also means there's less energy requirements. So, first of all, then let's look at why enzymes are specific. So, you should hopefully remember for GCSE the lock and key theory or the lock and key mechanism, and that is the idea that simply the substrate is like a key that fits into the lock of the active site, and they have a complementary shape to each other. So, the word shape there is really important, and basically that explains why enzymes are specific. So if you imagine that this enzyme here was protease digesting a protein molecule into amino acids, and then let's say what I'm drawing here was a lipid. Okay, if that was the case, the lipid wouldn't fit into the active site, and therefore the reaction wouldn't happen. If you were asked to describe the lock and key theory, you'd say about what I've just said. So you'd say the substrate acts as the key, it fits into the lock, which is the active site, which has the complementary shapes, so this bit here which fits together, okay, and they fit together to form, and you should always mention this term whenever you're talking about enzyme reactions. They fit together to form an enzyme substrate complex. The enzyme then will split the molecule, and it might be that the enzyme adds some pressure onto it, adds some force on it to pull the molecule apart, helping the hydrolysis reaction, and then the products have different shapes, so they no longer fit into the active site, so the products then leave as a result. So that's what the lock and key mechanism. You must mention that word, enzyme substrate complex. However, what has happened over time is we've sort of updated or upgraded this, so we don't just say the lock and key theory doesn't happen anymore, but it's like it's been advanced almost. So what we think now is that the molecules basically that fit in, the substrates still fit in to the active site, but the active site may not be a completely complementary shape. So it's a bit like if you had a glove or a sock. Let's say your sock has got a relative size shape to your feet, but it's only when your feet go into the sock that it actually fits sort of perfectly around your feet. And that is like what we're saying is happening to the active site here, is that the substrate fits in and the active site changes shape slightly to accommodate the substrate. And what happens is we have oppositely charged parts on the substrate and the active site. So where there is a charge on, let's say a positive charge on the substrate, so let's imagine that there's a plus charge here, so I'm just drawing on very basic, they would have an opposite charge, so maybe here, to hold it in place. And if let's say there's a negative charge here on the substrate, there'd be a positive charge there to hold it in place. But the idea is that the substrate wraps around, sorry, the active site wraps around and accommodates the substrate more. The changing shape of the enzyme then places stress on the substrate molecule, destabilizing it, allowing the reaction to occur easier, and that causes the lower activation energy. And obviously you get an enzyme, first of all, sorry, you get an enzyme substrate complex, so we should be mentioning that again, that term when they bind, that's what they form. And then it forms an enzyme product complex. Obviously the products have got a different shape, so then they're released and then the whole thing can be repeated again. Now, the evidence there is for the induced fit hypothesis is they've done this using X-ray diffraction. So that's the same way they figured out the structure of the DNA molecule. And basically fire X-rays off the enzymes to see at different points during a reaction what is happening. So what they would do is they'd look at this and look at the active site. So here you can see this is the active site of a molecule here, and the molecule is breaking down, so we converted glucose to glucose 6-phosphate. So where's that if you're doing year 13 work? That is a reaction that is involved in glycolysis to make the enzyme more, the glucose more reactive. So it's controlled by the reaction the enzyme hexokinase. So you can see here the hexokinase. You have before the glucose is added, and then when the glucose is added, you can see how the enzyme has changed its active site to suit the structure of the substrate. Now, there's still got the lock and key through there because the glucose would fit into the active site, but then it alters its shape to allow the reaction to happen. Um, so what else do we need to know? We need to know about the effect of heat energy, so temperature, on enzymes. So this is again very similar to GCSE, really, just a couple of extra points. So hopefully you understand that when molecules molecules constantly move due to the random motion, and that's due to kinetic energy. They constantly collide with each other as well. If you heat molecules up, they get more kinetic energy. That means they move faster and increases the number of collisions and also the force of collisions. There's no difference here between substrates and enzymes. The enzymes can only catalyze a reaction if the substrate collides with its active site, and that is random. They can't sort of suck them in, they have to randomly occur. So if you get increased temperature, I'm going to say here that a lot of enzymes are trapped in membranes, so they're not necessarily moving, but if you get an increased temperature, you get increased collisions because the substrates will be moving, possibly the enzymes, but the substrates will be moving quicker. You get an increased number of collisions, and then you get an increased rate of reaction, and that causes more products to be formed. What you get then is a graph that looks like this, which you should hopefully remember from GCSE. And again, like I've said before, you need to describe this graph both sides of that red line. So if you're describing about this side, which you must do and not forget, you would talk about the fact that the substrate is getting more kinetic energy, there's more collisions, that forms more enzyme substrate complexes, and that increases. And if there was a graph where you could read it off, you'd read some data off here, tell me what the different temperatures do. Then you get to your optimum temperature, which is where you form the most enzyme substrate complexes in most enzyme substrate complexes in a certain time, and that's your optimum temperature. Obviously, after the optimum temperature, the rate of reaction suddenly drops, and you must always remember to actually mention what the graph is showing. So the rate of reaction drops, easy mark. Why is it dropping? Because the enzyme is denaturing, which means its active site is changing shape, so it is no longer a complementary shape to the substrate, and that means you cannot form any more enzyme substrate complexes. Why does this happen? Because as the temperature increases, the particles that make the enzyme up or make up the amino acids even start to vibrate more and more. And at some point, they'll vibrate to the point where they start breaking the bonds holding the tertiary structure together. So what will start breaking first is probably your hydrogen bonds, and as a result of that, the tertiary structure starts to unravel and you will lose the shape of the active site. Therefore, the substrate can no longer fit in to the active site and you will no longer get enzyme substrate complexes. Don't forget that if it's cold, if it goes cold, that is a reversible change because it doesn't denature the enzyme, it just slows the collisions down so it can heat up again. But if you get denatured, it's permanent, and that means that you're not going to be able to recover afterwards because the enzymes have lost the shape of the active site. Now you can investigate this sort of experiments just to introduce one. You could look at maybe catalase against the enzyme I've been using as an example, and you need to know that it breaks down the enzyme catalase, which is in your liver, so it likes 37 degrees like the rest of our body, breaks down hydrogen peroxide into oxygen and water. So what you could do is you could place the following apparatus in water baths at different temperature. So we've got in here it's potato discs, and that's because whilst it's made in our liver, it's also present in potatoes and other plant materials because also plants produce hydrogen peroxide as a waste product that needs to be broken down. And effectively what we can do is we can use something called a manometer. And a manometer is basically where you have a test tube. You would have some trapped air in here. This would all be airtight at the top of the test tube, and you have a manometer. Now, a manometer basically is a tube that has coloured water in it down this bit in the bottom of the tube, and wherever the water stops, you would put that as your start mark, and there's ways you can calibrate that to do it. and you might time how long it takes the water to move up the scale to the 5 centimeters mark. So how long it takes to move up there. Now, why would it do that? Because one of the products is oxygen. So as this enzyme works, it will produce oxygen. That will increase the pressure, which will force more oxygen and air up through here, and that will push your coloured liquid round. And what you could do then is you can calculate by doing rate equals 1 divided by time. So if it took 60 seconds, you do 1 divided by 60. If it took 30 seconds, you do 1 divided by 30. And that would give you a comparable rate of this reaction. Just to go back to this point then, what is this? Well, this is actually a map of all of the enzyme reactions that are in your typical cells. There's only one bit that I'd expect you maybe to sort of recognize, and if you look at that, that's this bit, which is your glycolysis reaction linking in to the Krebs cycle, and that's only if you've done year 13 work as well. But this shows you how many enzymes are involved in keeping your cells together, and I'm guessing it's only an estimate as well. So hope that's been useful, guys, and I'll see you next time.